everyone, welcome to my channel Anupma Biology Classes. In this biotechnology series, today our topic is process of recombinant DNA technology. This video is taken from CBSE class 12th, part 3 of chapter 11. So let's come on the topic. Before this, in previous videos of biotechnology, we knew about principles of biotechnology and enzymes involved in recombinant DNA technology. Now, here you can see lecture overview that what we will see in this video. So in this video, we will see process of recombinant DNA technology, in which there are many steps like isolation of genetic material, that is DNA, cutting of DNA at specific locations, amplifications of gene of interest using PCR, insertion of recombinant DNA into the host cell or organism, obtaining the foreign gene product, and the last step is downstream processing. So in this video we will see all these steps, in which here first we will have to know that what are the steps of the recombinant DNA technology, in which there are many steps and these steps are carried out in a definite sequence. These are listed below. So in this, the first one, that is isolation of genetic material, that is DNA. So nucleic acid, that is DNA or RNA, is a genetic material of all organisms, and in most cases it is DNA, that is deoxyribonucleic acid. We also know that genes are located on DNA molecule. Thus, the initial step of our DNA technology is to obtain pure DNA molecule free from other macromolecules. In order to get pure DNA molecules, the following steps are required. So, bacterial cells or plants or animal tissues are treated with certain enzymes to break open the cell envelope. So in this picture you can see the lysozyme, which is bacterial cells are treated with lysozyme to dissolve the bacterial cell wall. Plant cells are treated with enzyme cellulase to dissolve cellulose cell wall, and the enzyme chitinase is used to dissolve fungal cell wall. This results in the release of DNA along with several other macromolecules and impurities such as RNA, proteins, polysaccharides, and lipids. Now, the next step is purification and separation of DNA. In this picture, you can see genomic DNA isolation and the DNA purification. The eukaryotic DNA molecules are intertwined with proteins such as histones. These proteins can be removed by treatment with enzyme proteases. The enzyme protease converts proteins into amino acids. Similarly, RNA are removed by treating with the enzyme ribonuclease. Other macromolecules can also be removed by specific treatments. Finally, the pure DNA molecules are precipitated out by adding chilled ethanol and collected in the suspension. So here you can see the two ways to concentrate the genomic DNA, that is spooling and ethanol precipitation. Now, the second step of recombinant DNA technology formation is cutting off DNA at a specific location. So, cutting off DNA at specific locations by agarose gel electrophoresis DNA undergoes restriction enzyme digestion, or cutting off gene of interest and addition of ligase. Then recombinant DNA is formed. So these are the steps after the cutting of DNA at a specific location. The cutting of DNA at specific locations became possible with the discovery of probes, selectable markers, ligases, and restriction enzymes. Isolated and purified DNA molecules are cleaved by using the enzyme restriction endonuclease. This enzyme The action of enzyme restriction endonuclease has been discussed in detail in earlier videos. You knew that what is restriction endonucleases? Is restriction endonucleases cut DNA at specific sites. As stated earlier, the enzyme restriction endonucleases cleave DNA at a specific site, resulting in the formation of sticky ends. In practice, the digestion by a restriction enzyme is performed by incubating the purified DNA molecule with the specific enzyme, keeping all other conditions at the optimum level. The progress of digestion is checked by using agarose gel electrophoresis. As the DNA molecules are negatively charged, they move towards the positive electrodes. The same process is repeated with the vector DNA. The recombinant DNA, as stated earlier, is the DNA formed by combining DNAs from two different organisms. In this case, here you can see the enzyme cuts both DNA strands at the same site. That is, first is vector DNA and the second is foreign DNA. So, in this case, the purified and cleaved DNA molecules, that is both the source DNA as well as the vector DNA, are mixed. More specifically, the cutout gene of interest from the source DNA is mixed with the cut vector, that is having appropriate space. These two are mixed in presence of enzyme ligase so that their sticky ends join, resulting in the formation of recombinant DNA. So here you can see that how the enzyme cut both the strands at the same site, and then the DNA fragments join at sticky ends, and after that recombinant DNA formation. So these are the steps in the formation of recombinant DNA by the action of restriction endonuclease enzyme ECOLOGY. Now the third step is here amplification of gene of interest using PCR. So, polymerase chain reaction, that is PCR, is the technique of synthesizing multiple copies of the desired gene or DNA in vitro. This technique was developed by Carey Mullius in 1985. Now, this technique here, it is based on the principle that a DNA molecule, when subjected to high temperature, splits into two strands due to denaturation. These single-stranded molecules are then converted to original double-stranded molecules by synthesizing new strands in presence of enzyme DNA polymerase. Thus, a double-stranded molecule of DNA is duplicated and multiple copies of the original DNA sequence can be generated by repeating the process several times. Now, the basic requirements of the PCR. So, there are three basic requirements, in which the first one is DNA template. The desired segment of the target DNA molecule that is to be amplified. Second is two nucleotide primers. Two nucleotide primers, usually 10 to 18 nucleotide long and complementary to the sequences present at the third end of the target DNA segment. And the third is enzyme. So, high temperature, more than 90 degrees Celsius, is stable DNA polymerase, usually Taq polymerase, for synthesis of new DNA molecules are required. Now here you can see the working mechanism of PCR. So in the mechanism of PCR there are three steps, that is denature, anneal primers and replicate. First of all, the target DNA, that is DNA segment to be amplified, is heated to high temperature, that is 94 degrees Celsius. Heating results in the separation of two strands of DNA. Each of the two strands of the target DNA now act as template for synthesis of new DNA strand. This step is called denaturation. Denaturation is the separation of the two strands of DNA by breakdown of hydrogen bonds on heating, which you can see in the picture. That is what is the process of denaturation. Denaturation is followed by annealing. Annealing means Anneal means join. So during this step, two oligonucleotide primers hybridize to each of single-stranded template, that is DNA, in presence of excess of synthetic oligonucleotide. Annealing is carried out at lower temperature, that is 40 to 60 degrees Celsius. Third and final step is extension. Here in this picture you can see that the first is denaturation, then annealing, and after that the extension. During this step, the enzyme DNA polymerase synthesizes the DNA segment between the primers. Usually, Taq DNA polymerase isolated from a thermophilic bacterium, Thermus aquaticus, is used in most of the cases. The two primers extend towards each other in order to copy the DNA segment tying between the two primers. This step requires presence of deoxyribonucleoside triphosphates, that is DNTPs, and magnesium ion, and occurs at 72 degrees Celsius. So, the mentioned three steps complete the first cycle of PCR. That is, here you can see 95 degrees Celsius denaturation occur, then 50 to 70 degrees Celsius primer annealing, then on 72 degrees Celsius polymerase extension, then new double-stranded DNA template formation. So the second cycle begins with the denaturation of extension product of the first cycle, and after completing the extension step, two cycles are completed. If these cycles are repeated many times, the DNA segments can be amplified to approximately billion times, that is 1 billion copies of desired DNA segments are made. If required, the amplified fragments can be ligated with a vector for further cloning. So, this is the complete process of amplification. Now, the fourth step is here, that is insertion of recombinant DNA into the host cell or organism. So, here you can see techniques of genetic modifications, that is inserting foreign DNA into the cells. So there are many steps, that is DNA can be inserted into a cell by transformation, protoplast fusion, electroporation, and microinjection. Several techniques, that is vector mediated or vectorless gene transfer techniques, are used to introduce the ligated DNA into recipient cells. Once a cell receives a foreign DNA fragment and becomes transformed into a genetically modified cell, it starts behaving differently. For example, if a rDNA bearing gene which is resistant to antibiotic ampicillin is inserted in E. coli cells, the bacteria become resistant to ampicillin. So here you can see the process of insertion, that is plasmid and human cell, then recombinant DNA formation, then the transgenic bacterium with plasmid containing insulin gene, then the recombinant bacteria gain ability to secrete human insulin. This is the main process of recombinant DNA formation, that how a vector DNA and the foreign DNA introduced and form a new recombinant DNA. If such bacteria are transferred on a culture plate containing the antibiotic ampicillin, only the resistant forms will grow and other will die. This method can be used to select out the transformed cells from a collection of bacteria. The gene responsible for ampicillin resistance is called a selectable marker, which you can see in this picture. So what is selective marker? Selective marker is required for maintenance of plasmid in the cell. Because of the presence of the selective marker, the plasmid becomes useful for the cell. Under the selective conditions, only cells that contain plasmids with selectable marker can survive. Genes that confer resistance to various antibiotics are used. Genes that make cells resistant to ampicillin, neomycin, or chloramphenicol are used. So this is all about the insertion of recombinant DNA into the host cell or organism. Now, the fifth step is obtaining the foreign gene product. So what is this? When a piece of foreign DNA is inserted into a cloning vector and then transferred into a bacterial, plant or animal cells, it gets established. After establishing the recombinant DNA inside the host cell, they are allowed to express their role in the synthesis of desired proteins. Such proteins formed from recombinant DNA in a heterologous host are called recombinant proteins. In fact, most of the recombinant DNA technologies are aimed to produce desirable proteins. Therefore, such cells having recombinant DNA are grown on a small scale in the laboratory under controlled conditions. They are allowed to synthesize the desired proteins, which are finally extracted and purified from cultures. Alternatively, the host cells are multiplied in a continuous culture where the used medium is allowed to pass out from one side and fresh medium is continuously added into the culture vessel from the other side. It maintains the cells in their physiologically most active lag exponential phase. So here you can see that is transformation of competent E. coli with the gene of interest, then selection and expansion of positive clones, then isolation of plasmid expression vector, then scale up of transformants, isolation of proteins of interest. Small volumes cultures are usually employed in laboratories for research and production of less quantities of products. So, however, large scale production of the product is carried out in the bioreactors, which you can see in this picture. This is a simple stirred tank bioreactor. Bioreactors are large vessels having a volume of 100 to 1000 liters, which are used for biological conversion of raw materials into specific products. Each bioreactor has a cylindrical stirred tank to facilitate the mixing of contents. The stirrer provides facility of mixing the contents as well as the availability of oxygen. A bioreactor is more advantageous than sick flasks. It has an agitator system to mix the contents properly, an oxygen delivery system to make availability of oxygen, a foam control system, a temperature control system, a pH control system, and a sampling port to withdraw the small volumes of the culture periodically. Now, applications of the bioreactors. So, what are the applications of the bioreactors? Bioreactors are used for biological conversion of raw materials into specific products. These are used for processing large volume of culture. These are used for the production of larger biomass leading to higher yield of desired proteins. So these are the applications of bioreactor. Now, after this, the last step is downstream processing. So, once the foreign gene product is synthesized, it is collected from the bioreactor and then processed further for its separation and purification. The purified and finally finished product is now ready for marketing. After biosynthetic stage, the collection of product, its separation and purification is known as downstream processing. Before marketing, the product has been formulated with suitable preservatives. In case of drugs obtained from recombinant proteins, clinical trials and strict quality control testing must be carried. These quality control testing and clinical trials vary from product to product. So in the process of downstream processing, here you can see that is intracellular products, then cell disruption occur, then removals of the insoluble, and here the extracellular products, and then isolation, then purification, then finishing, and then the final product obtained. So the complete stages of the downstream processing are removal of insolubles, then product isolation, then product purification, then product polishing, and the last is to markets. So this is the end of video in which you knew about the process or steps of recombinant DNA technology. The next upcoming video in biotechnology series will be on the transgenic organism. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. Keep watching, keep improving, keep learning and subscribe to my channel. Thank you for the watching.